Cells and NLRP3 expression. I've talked about cells quite a few times, especially white blood cells like macrophages, monocytes and neutrophils. This is because those cells express NLRP3 and are therefore important for CAPS, which is an autoinflammatory disease caused by hyperactive NLRP3. When I say cells express NLRP3, what I mean is that the NLRP3 gene in our DNA is actively transcribed into messenger RNA that is then converted into an NLRP3 protein. We have over 200 different types of cells in our bodies, and they don't all express the same genes. Putting that another way, different cell types express different genes, and it makes sense, right, that cells in your liver aren't going to express the same genes as, say, neurons in your brain, because they perform very different functions. But NLRP3 is expressed in a lot more cell types than just macrophages, monocytes, and neutrophils. Lately, I've been trying to work out which cells do express NLRP3, and from various sources so far, I have found lymphocytes, which are B and T cells in your adaptive immune system, chondrocytes, which are cartilage cells, epithelial cells, which are cells that line outer surfaces of things like skin and internal organs, osteoblasts, which are a type of bone cell, microglia that are considered to be the macrophage equivalent in the brain, and granulocytes, which include neutrophils but also eosinophils and basophils, which are other white blood cell types. There are more, but this area of study is frankly still really vague and needs a lot more work. So I've had this question rolling around in my head for a while now. Why do people with CAPS get different symptoms? The rash is almost universal, almost. But one of the symptoms that defines Muckle-Wells syndrome from familial cold autoinflammatory syndrome is hearing loss. I have A439V mutation. That puts me across the two conditions, but I don't fit neatly into either one. Without medication, I do get cold-triggered episodes that are more like FCAS, but I get daily symptoms that are more like Muckle-Wells in frequency and severity. I get killer migraines and I get gastrointestinal complications which are associated more with Muckle-Wells syndrome, but I don't have hearing loss. So for starters, I specifically want to know, why do some people with CAPS have hearing loss and some don't? And why do some people with CAPS have gastrointestinal complications and some don't? Do these symptom differences have anything to do with NLRP3 expression in different cell types? And do the different mutations have anything to do with influencing the NLRP3 expression in those different cell types? Starting with the hearing loss question, a paper in 2020 found that the NLRP3 inflammasome can be activated in inner ear macrophage cells. They described an interesting NLRP3 mutation of R918Q with hearing loss as a primary symptom and being quite different from classic CAPS. The researchers wondered whether the hearing loss is secondary to systemic inflammation with cochlear infiltration of circulating immune cells, or primary autoinflammation with pathologic activation of the NLRP3 inflammasome within the resident immune cells of the cochlea. They found it was the tissue-resident cells. This was in contrast to a mouse model study from Korea this year that worked with the equivalent of the CAPS mutation D303N, and they found that both the cochlea infiltrating and the resident macrophages contributed to the hearing loss. But D303N is a more typical CAPS mutation, and that's worth noting. So again, I wonder, do the specific mutations somehow contribute to the localization of NLRP3 expression? We know that NLRP3 forms an inflammasome. It gets together with other NLRP3 proteins and it needs energy from ATP to do this. The grouping of NLRP3, the fancy word is oligomerization. It draws in the adapted protein ASC, and that in turn recruits caspase-1, which activates itself, and then that activates interleukin-1 beta, interleukin-18 and Gasdermin D. We have also recently learned that NEC7 and NLRP11 are inflammasome components too. So we need to find out if all the other inflammasome components are expressed in all of the cell types that express NLRP3 and how they might be regulated and how they interact with NLRP3 exactly, so we can figure out how the different mutations might be affecting those interactions. It's a lot of work to do. Let's say, for example, hypothetically, say immune cells in the gut or epithelial cells in the gut can form an inflammasome but don't express interleukin-1 beta. And say that the A439V mutation causes constitutive activation by ATP binding, which is necessary in those particular gut cells. But without interleukin 1 beta, all that gets activated by the inflammasome is interleukin 18, another cytokine that has been described as protective in acute gut inflammation, but damaging in chronic long-term gut inflammation. And then say again, hypothetically, say another mutation, T348M, causes NLRP3 to be activated by enhanced NEK7 binding. It makes it less hyperactive in the gut cells, but more hyperactive in chondrocytes, cartilage cells in the ear. Maybe that triggers the resident cochlear macrophages and causes the hearing loss. And say the A439V mutated NLRP3 is less active there, so we find less hearing loss with that mutation generally. Right now, I'm purely speculating with this thinking. This is the kind of speculation that would have been fun to do with my old man, who loved a bit of blue sky thinking. Like me, he had gastrointestinal trouble, which went from mouth ulcers to reflux to all the digestive discomfort, abdominal pain, and the other end business that nobody likes to talk about. But suffice to say, it's dysfunctional and not quite your average inflammatory bowel disease. I know that his mum, my grandma, had problems like these too. And in patient network groups, this stuff is common among other patients who have the same mutation as me. And there are other mutations that seem to pop up consistently with gastrointestinal problems, like the D303N that I mentioned, R260W, and some of the other mutations considered not to be classic CAPS, like V198M, Q703K, and R488K, where it appears to be one of the primary symptoms. So there's definitely something here worth investigating.